Hello, my name is Dale. In another video, I provided information about my company and its platform technology, BRG Therapeutics. Here, I wish to provide information about our understanding of the mechanisms that underlie the effects of these drugs. Note, BRG Therapeutics are in the preclinical stage of development and still require further testing and regulatory review. Every year, millions of people are affected by cancers and viral infections, and millions of people die due to these diseases. What if we could make a significant impact on these conditions? BRG Therapeutics offer this possibility. How do they do this? To better understand BRG drug mechanisms, we have taken data from our published peer-reviewed paper and combined that with information from several other pertinent sources. This process led us to 3 probable major modes of action presented here. Here, I will focus on p53 protein and its circuitry because of its importance in health, disease, and especially in cancer. This is an image of the p53 protein and its circuitry. This is simplified and only shows a few highlights. The actual circuitry is much more complex with many more components. But I'm showing several important things that this circuitry does. In particular, it is able to provide protection to normal cells while also removing damaged cells like cancer cells and virus-infected cells. And the net outcome of its activity is the preservation of cellular and genomic integrity. I'm going to focus on cancer. Approximately 50% of all cancers have mutations in the p53 gene. This creates a block in the circuitry, and that allows cancer cells to survive and proliferate. The other 50% of cancer cells have other mechanisms that they can use to avoid p53 effects. They can have defects in upstream pathways that result in signaling that bypasses the effects of the p53 circuitry and allows cancer cells to survive. Another mechanism used by cancer cells with normal p53 protein is to have defects in downstream components that receive signals from the p53 circuitry. Once again, this introduces a block and cancer cells can survive. Now, most of us know if we break something, it doesn't work. But that's not the case with the p53 circuitry. For a subset of p53 mutants, they lose normal function, but they gain abnormal function. This is called gain of function, and it plays a major role in cancer cell survival and drug resistance. So how do BRG active ingredients affect the p53 circuitry? Here's one example. BRG active ingredient number 1 is reported to restore normal p53 function for a substantial portion of mutant proteins. This removes the block and allows damaged cells to be removed. Both active ingredients show evidence of being able to block signaling from abnormal upstream pathways that allow cancer cells to bypass the effects of p53 circuitry and survive. These active ingredients also show evidence of being able to restore normal function to p53 circuitry downstream pathways and processes so that cancer cells can die. Our preclinical study results also support the ability of BRG active ingredients to prevent mutant p53 gain of function effects. Viruses also use the p53 circuitry for their replication. This has been shown for multiple viruses, including influenza and HIV, the virus that causes AIDS. This is suspected for most or all viruses. So, in summary, through their effects upon the p53 circuitry and other mechanisms not discussed here, BRG Therapeutics have the potential to provide a significant beneficial impact upon cancer and viral infections. So invest with us. Let's work together to advance this family of drugs and improve the outlook for these diseases. Thank you.